okay?
Yes.
Yeah.
Hello, my name is Alexander Fedintsev. I'm a Chief Science Officer at Radical Life Extension Group. Our group is a non-profit based in Spain, and we are aiming to achieve radical longevity within our lifetime. In 2020, I proposed 10th hallmark of aging, which is stochastic, non-enzymatic damage to long-lived macromolecules. And it seems it is time for yet another one hallmark of aging. But before, next slide. Yeah. But before that, I want to talk a little bit about radical longevity and especially about longevity escape velocity. This concept, if I'm not mistaken, was introduced by Aubrey himself back in the days. And basically, it means that technological progress every year, on average, adds additional year of life expectancy. I prefer to call, by the way, this concept death escape velocity because I think it is more clearly conveys the message. And I decided to check how far are we from death escape velocity. And I pulled some data across European countries and fitted the regression lines. And it seems that every year, technological progress gives us a quarter of a year. Well, that's not bad. That's a pretty good start because, well, it increases the life expectancy of people that are born nowadays by a third, approximately. But unfortunately, these numbers are about total life expectancy at birth. And if we look at the life expectancy at 85 plus years, or like the dynamics of the lifespan increase, we see much smaller increases across multiple countries. On average, we are talking about 1/10 of a year per year, and this is almost like a negligible thing. So it means that our median lifespan increases, but the maximum longevity, maximum, of course, is not a correct term here, but anyway, it virtually stays the same, which is a pretty bad picture. And why this discrepancy is there? The answer to this conundrum remains elusive. But today I will present one thing that I think might play a substantial role here in this discrepancy. And I am talking about a thing that is called clonal hematopoiesis, or clonal hematopoiesis of indeterminate potential. So hematologists, blood doctors, they are aware of this for quite a long time, but it wasn't very known in the biogerontology community. At least I've never heard about it in a wider scope. Like, and what is it? What is the chip? We have blood stem cells. Also, they are called hematopoietic stem cells that give rise to virtually all blood cells in our body: red blood cells, lymphocytes, neutrophils, platelets, whatnot. And they proliferate, and of course, they accumulate mutations. And unfortunately, some of the mutations are giving certain traits to hematopoietic stem cells. And certain mutations increase their self-renewal. But unfortunately, it is a double-edged sword because these mutations also impair the ability to produce healthy blood cells. And this is what actually happens with age. So these mutants, these clones, because of their increased capacities to self-renewal, they outcompete healthy cells, and our blood is full of damaged descendants of these stem cells. Why is it important in context of lifespan increase? Well, because it grows almost exponentially with age. So like a person who lived till like 100 or past 100 years will almost surely have clonal hematopoiesis. And it is like a huge body of evidence suggests that clonal hematopoiesis leads to an increase of cardiovascular disease rates because of multiple mechanisms. But one of the main mechanisms is so-called myeloid bias. So many mutations that we see during clonal hematopoiesis CHIP, they basically shift hematopoiesis towards the myeloid cells and not lymphoid. Lymphoid cells are responsible for adaptive immunity for cancer, fighting viral infections, while myeloid cells, they are representing innate immunity, and they are extremely pro-inflammatory. And this could be one of the leading causes of inflammaging. Like if we look at the data, so we can see that like in the bottom line, CHIP present means that there is like a clonal hematopoiesis. It increases cardiovascular mortality, or like I'm not sure whether this is mortality or disease incidence, but anyway, it increases like by 80%. And we see that the degree of clonal hematopoiesis is associated with cumulative incidence of atherosclerosis. So no CHIP is a green line, small size of a clone is violet line, and then big clone is red line, and it is extremely detrimental for survival. But not only clonal hematopoiesis is associated with cardiovascular diseases. It is also associated with cancer and other malignancies. The thing is that, in fact, CHIP is a pre-cancerous thing, and it increases the risk of blood cancers tenfold. But not only that, it increases also like risk of lung cancer by like 60%. And then there was another study that kind of shed light on the causative relationship between CHIP and cancer, because mice, murine model of colon cancer, like showed that if we inject clones into the bone marrow of mice, it exacerbates colon cancer rates and decreases survival dramatically. And also, clonal hematopoiesis is associated with not so deadly, but still very, very bad consequences of aging, such as osteoporosis and arthritis, so which is quite important for the quality of life. And I'm pretty sure that there is more. It is just so understudied. But anyway, what causes CHIP, right? Why it occurs? Well, unfortunately, it's inevitable because CHIP is caused by mutations. Mutations occur randomly, like anyway, right? So otherwise we wouldn't have cancers and whatnot. But some conditions give these clones with certain mutations advantage to outcompete healthy cells. And it seems that the increased inflammation is the key. So these clones proliferate faster in inflammatory conditions. And this is very important. And we kind of see a self-increasing vicious cycle. So certain mutations like increase inflammation, and the increased inflammation give a boost to these pathogenic clones. And here is one connection between the hallmark that I proposed and the chip, because glycation alters the collagen and other long-lived molecules, and it basically stimulates RAGE, receptor for advanced glycation end products, and increases inflammation. And this constant inflammation might play a role in survival of these pathogenic clones. Interesting connection between CHIP and epigenetic age. The thing is that the most common mutations during CHIP happens in genes that are associated with methylation. DNMT3A and TET2 are demethylase and methylase accordingly. And well, it's not surprising that having CHIP is associated with higher epigenetic age. Well, having like multiple mutations, multiple clones with different mutations is associated with epigenetic age that is 7.4 years older than chronological age. So clonal hematopoiesis is, in my opinion, one manifestation of a more generic phenomenon, clonal expansion. Right? So when some clones in some environments get an advantage and outcompete healthy cells. For instance, this happens within atherosclerotic plaques. So smooth muscle-derived cells within atherosclerotic plaques have specific mutations, and they proliferate, and this what causes atherosclerotic plaque to grow. And scientists in this amazing study, they showed that using an oncology drug, niraparib, kind of ameliorates the atherosclerosis in mice. And they proposed to like a new area of medicine, athero-oncology. Another thing is very interesting study that showed that mesenchymal stem cells, they have distinct population within the pool of mesenchymal stem cells. And the youthful stem cells, they are still there in the pool of stem cells of older donors, but they are reduced in quantity. But if we isolate them and grow on the ECM media, they expand like several billion times and are perfectly healthy. So the thing is that there is some clone that, or multiple clones that outcompete them and kind of suppress normal healthy cells. But this gives us like a hope that we can restore the quality of our stem cells just by suppressing or killing these pathogenic clones. Okay, before solving the problems, we first need to measure the degree of problems. How we detect and measure CHIP. Well, the best method, the most reliable method is, of course, full genome sequencing with high coverage, because we can see then like what are the variant allele frequency. Well, basically, roughly speaking, it means like how many, what is the fraction of a clone in the peripheral blood cells. But it is costly and not routinely available, so we probably want some markers that kind of narrow down the scope. And there are some good markers. For instance, this study identified that neutrophils and RDW, red distribution width, red cell distribution width, are strongly positively associated with CHIP. Another study showed that that patients with CHIP have lower lymphocyte, like lower counts of lymphocytes and higher counts of neutrophils, which kind of in line with this myeloid bias. Pretty unsurprising. And my favorite biomarker is RDW. What is it? It's a very, very simple, routinely available biomarker that is strongly associated with mortality, disease incidence, and whatnot. And there are some evidence that it is associated with CHIP. So what is it? It's just a variation in the volume of erythrocytes, red blood cells. So the higher the variance, the higher the variability in size, the more likely one has a CHIP, clonal hematopoiesis. We did some data. We have a huge database of blood analysis, blood samples, and we see clearly on the left panel that RDV increases strongly, like with age, and also it is negatively associated with red blood cell count, which might indicate that there is a negative impact of clonal hematopoiesis on our erythrocytes. But this is like a very preliminary hypothesis. All right, how we combat clonal hematopoiesis? Well, one good strategy is just to kill the clone or kill the myeloid precursors. And this actually, in this paper, it was shown to rejuvenate immune system in mice, in murine model. And so in this study, investigators, they've identified surface proteins that are enriched in myeloid-based precursors. After identifying certain proteins, they created specific antibodies that target and eliminate these myeloid-biased stem cells. And, well, basically it led to sufficient, substantial rejuvenation of several markers of immune system. I think this is a viable approach, though it sounds like a little bit dangerous to me to kill cells in apparently almost healthy adult, right? So if it's a cancer patient, it might be worth it to kill cells, but yeah, in almost healthy older adult, killing cells sounds like a little bit of an overkill to me. But there is another strategy that I think is more suitable and closer to practice. This study in rhesus monkeys, if I'm not mistaken, is really a breakthrough in my opinion. First, scientists showed that clonal hematopoiesis occurs naturally in monkeys, which is not the case for mice, for instance. And second, they've created, they've induced clonal hematopoiesis similar to what we humans develop with mutations in DMT3A and TAT2. And then they showed that inhibition of a certain inflammatory cytokine, IL-6, results in slowing down of the growth of this clone. But unfortunately, the downside of it, so we see that in some monkeys, like in the first monkey, the clone actually started to shrink because the growth rate was negative. In other, there was just a slowdown of the growth, but it's still something. Unfortunately, after cessation of tocilizumab administration, tocilizumab is a monoclonal antibody that blocks IL-6. We see that the clone rebounded, so it starts to grow again. So in this case, we need a lifelong administration of anti-inflammatory drugs. And that's why we need something that is economically viable, right? Because like monoclonal antibodies cost a lot, and cost efficiency is not that great. But anyway, I think this is the best possible strategy that we can use right now, because there is also evidence from humans. There are people with a mutation in IL-6 gene that kind of inactivates this gene, and when we look at the chip carriers and we stratify by IL-6 mutation, we see that people with mutation and chip, they have like much lower cardiovascular disease mortality than those who have chip and have normal IL-6. So I think IL-6 inhibition is basically the best available strategy to at least slow down clonal hematopoiesis. And this is what we currently are experimenting with on humans. On human volunteers, we are trying to slow down clonal hematopoiesis. A couple of words about our group. We are a Spain-based nonprofit aimed at bringing or achieving radical life extension within our lifetime. We probably were the first who tried therapeutic plasma exchange on humans when it was like in 2020, I guess. And since that time, we switched to more promising interventions, like we're developing gene delivery platform, and now we work also on clonal hematopoiesis. So what we did, we identified a person who likely has a clonal hematopoiesis. So we see that this person has a huge amount of neutrophils, low lymphocytes, very high C-reactive protein, which is an inflammatory marker, in absence of any known viral and bacterial infections, whatnot. Also, RDW was pretty high, and IL-6 obviously is much higher than in normal persons. And we confirmed, by sequencing the genome, we confirmed the presence of TAT2 clonal hematopoiesis. And we started experimenting with different small molecules, because we can, of course, go the way that was shown in the monkey paper, right? So administering tocilizumab. But tocilizumab has a lot of unwanted effects because it blocks both classic and trans signaling of IL-6 and creates a lot of adverse effects. So we wanted first to experiment with small molecules, and the cocktail of small molecules, all are FDA-approved drugs, so all are kind of safe and tolerable, and we achieved this dynamics of inflammation in the body. And so we haven't reached our milestone yet, so we want to suppress inflammation completely or make it undetectable. So we're still experimenting with parts of this therapeutic cocktail with ingredients. And we are monitoring now different markers and, of course, the clonal hematopoiesis. So basically, it will be a long journey because we are not expecting immediate results. And we think we will be able to at least slow down clonal hematopoiesis. But then what I urge you to basically pay more attention to this very important age-related condition. Thank you for your attention.
Does anybody have any questions at all for Alexander? We have Sebastian over there, so if we can just get the microphone to you, Sebastian, please hold on.
Hi, Alexander. Thank you for
Hi.
the presentation.
Thank
this
you.
is this review is a helpful support for ImmuneAge Bio, where we're rejuvenating the hematopoietic stem cells. And actually, some of the molecules we found support some of your hypothesis about inflammatory cytokines. So we found that reducing IL-6 and TNF-alpha can rejuvenate HSCs. I'm curious, you brought up the theory about atherosclerosis being driven in large part by systemic aging driven by aging bone marrow. And you talked a bit about this sort of tumor-like formation in the vascular epithelium. So can you elaborate a little bit on the mechanism that you think is driving that?
Yeah, sure. So, well, the mechanism is basically the same that, like evolution. Cells, for instance, atherosclerotic plaque has high oxidative stress levels within the plaque, so which kind of increases the mutation rates. And then, but mutations are random, right, basically. But if we have a specific conditions, so which favor some certain mutations, right, then we can have a clonal expansion. So some mutations some cells with certain mutations get an advantage and start to proliferate better, and they outcompete other cells. So this is how we get clonal expansion.
Yep, we have another question at the front.
One of the things I look at clinically is the lymphocyte to neutrophil ratio.
Yes.
And as that changes, basically as the neutrophils go up and lymphocytes go down, and I get a patient, if it's a viral infection, bacterial infection, I'm like, oh no. How much of that, do you have a sense of how much of that is due to these chip cells? Is it all due to these chip cells? Is it partially? I'm just kind of wondering how much these are related.
If it's a persistent phenomenon, then it is likely due to some imbalance between myeloid-biased and balanced hematopoietic stem cells precursors. But if it's temporary, it might indicate that there is like some viral infection that depletes a lymphocyte pool or something like that. So, but if it's like persistent, like over years, then it might indicate chip.
Great. Thank you.
Hi.
Another question. Thank you.
So great talk. But I'm really intrigued. So IL-6 has evolved to be able to have a protective effect when you have an infectious disease.
Yes.
So lifelong inhibition of IL-6 may limit this age-related inflammation, but how are the patients doing if they're infected with a virus, for example?
Yeah, it's a very good question. But we have here we have a good example, because we have like a good thing to refer is like patients with rheumatoid arthritis, right? And then some of them are taking IL-6 inhibitors throughout like many decades. Maybe not many, but more than a decade probably. And actually, there is some increase in infections, but it's not dramatic. And there is also a decrease in cardiovascular disease mortality. So we have a net gain here in life expectancy. But I think the future is towards more specific inhibition, because like current IL-6 inhibitors, they are not ideal because they, as I've mentioned, they inhibit like both classic and trans signaling mechanisms. And one of them is like classic is so to speak good, and trans signaling is bad, like pro-inflammatory and so on and so forth. And that's why many unwanted effects occur. And if we can more precisely target only pro-inflammatory pathway, we will be in a much better position. And there are drugs that are in development nowadays for exactly rheumatoid arthritis that basically are much safer. But of course, they're also very expensive.
Is there a solid evidence that the skewing towards myeloid is not a compensatory mechanism?
Well, it's hard to say, but as we saw in the paper, so eliminating these myeloid cells, well, improves like rejuvenates the immune system, and from cross-sectional data we have like knowledge that this, as a person mentioned, like neutrophil to lymphocyte ratio, if it's increased, then the prognosis for many diseases is worse. And so I don't think it's an adaptation. I think it's a maladaptation.
And like, so the strategy that you propose is reducing the myeloid lineage. But what about in the other strategy, which could be increasing the lymphocytes?
Well, it's not easy to do that, because like the lymphocyte producing cells are reduced in quantity. Yeah, so we can of course boost them, but for that we need to suppress the clone that kind of gain like advantage over lymphocyte producing stem cells. So that is the main point. So you can't just increase lymphocyte production because without like suppressing this bad myeloid biased clones, because they kind of they hijacked all the space, living space, so to speak, in the bone marrow.
Thanks.
Great. Alexander, thank you so much.
Thank you.
I knew you'd get some questions, so that's wonderful. Thanks very much for joining us today. Please, thank you.